Okay, anyway, that's a good time. People had a coffee, and this break, I think, is very good after all the excitement. You know, well, first, I want to thank the Bankinter Foundation for inviting me to come. I used to be a trustee a few years ago, and it's always such a pleasure to meet these people again and to get to meet new people and to participate in these amazing discussions that we had today. So congratulations already for the organization. And so I'm going to touch a subject that was touched already this afternoon, but in a different way. I mean, we have been talking a lot about human enhancement, about RNA, about gene editing, and I would say that all the discoveries around RNA and human enhancement have somehow unveiled new horizons in my field of research, which is nanotechnology applied to medicine, nanomedicine. So I know we have been discussing about what is the right definition of human enhancement, but in a very broad way. And I had a discussion with some key people like Robert Langer and others in the field before coming here. What do you think it is human enhancement? RNA delivery can fit there. And well, he said, well, you know, it depends in which sense you want to take this idea, this concept. In a generic way, human enhancement means that we are aiming at improving human traits, abilities, or functions. And indeed, the causes of diseases are biological dysfunctions. Very often, these dysfunctions are related to genes which do not work in the right way, which are anomalous. And they are producing proteins, encoding proteins that either are aberrant, there are too many, the production is too high, and this leads to a disease, or either the production is not sufficient. In the majority of cases, diseases are due to this. Erroneous proteins, too many proteins, or too little amount of proteins. And now we are facing, as Dr. Collin was saying today, we are facing an amazing time in medicine, in therapies. It is indeed a paradigmatic change in medicine because, for the first time, we are confronting not the symptoms of the disease, but the reason, the cause of the disease. And we are letting our body to repair itself. We are not repairing it. We are giving the body the information to repair itself. And this is a dramatic change in medicine. So taking this subject in a very easy, generic way, I'm not going to talk about gene editing, although it is strictly related, but I'm going to talk about more simple therapies, which are not in the future. They are already there, and of course, the future is very promising. So in a very broad way, we can make two categories in RNA technologies and RNA uses in medicine. We have what everybody knows, mRNA vaccines, and as you can see, well, producing RNA is very easy. People may think that, you know, it's complicated, but in fact, we produce RNA from DNA, and then we administer the RNA with the information to our body, and our body produces the protein of interest. And this is the principle of the vaccines. We are not receiving a vaccine. Actually, we are receiving the genetic information for the production of the vaccine in our body. And this is the case of COVID vaccines. On the other hand, we have RNA-based therapies. And as I said before, we are dealing with the origin, the cause of the disease. And there are two main ways. There are many ways, but in a simple approach, we could say there are two ways to deal with the disease. We can silence a gene which is responsible for the production of a defective protein, and we silence it. We can do it. And on the other hand, the other option is to administer, as in the case of the vaccines, an RNA that has the information, so our body produces the protein that we need in order to cure the disease. So what are the main technologies involved in these kind of therapies and vaccination? Because today we have been hearing a lot about RNA, about artificial intelligence, about omics, and actually all these technologies have to be integrated together. Of course, we need proteomics, genomics to identify the genes that are erroneous. We need biotechnology in order to produce RNA, DNA, RNA proteins that can be used as treatments. And we have so much information from omics and from biotechnology that we need to use artificial intelligence to better design these kind of treatments. But something that people forget about, and I was very pleased when Dr. Collin was saying today that the problem may be for gene editing the delivery, and this is where we are. We are working on nanotechnology. We are making nanoparticles. There is not a pointer here, right? No. No problem. It's very clear. So you can see there a couple of nanostructures that were made in our lab, and you can see that they are made of different materials with different morphologies. And in fact, nobody has been talking about them, but these kind of nanoparticles, nanocarriers, have been essential for the vaccination, for transporting the RNA to the right place, and for producing the antigen. So just to give you an idea about what nano means, a molecule of a protein, such as insulin, is around a few nanometers. The size of a virus is around 100 nanometers, and actually the size of the particles we prepare is kind of artificial viruses, so yes, 100 nanometers. They are much smaller than a bacteria, and of course, much smaller than a cell. As a matter of fact, if we could somehow translate the size, you could see that in one nanoparticle, we can pack tens or hundreds of molecules of drugs. A number of particles can go into a bacteria, and of course, hundreds of particles can get into the cell transporting the RNA. So if you remember, when we had the COVID vaccines, people were saying, how is that possible in such a short way? Some people were even suspicious about whether the vaccine was going to work, was going to be safe. But what people did not realize is that researchers have been working in these fields since the 60s. As you can see in the slide, for example, the discovery of mRNA, the function of mRNA was in the 60s. At the same time, were produced the first lipid nanoparticles. We used to call them liposomes. Lipid nanoparticles and liposomes is pretty much the same, but people now call them lipid nanoparticles. So it was already in the 70s when we had the first liposomes or lipid nanoparticles loaded with mRNA, and in the 90s, we already had vaccines, RNA formulations for influenza vaccine. So, over the last decades, we have been obviously evolving. I'm not giving the details, but many advances have been produced in the last decades, so that in 2014, we already had formulations containing mRNA for immunotherapy, and a few years later, we already have some vaccines in clinical trials. Obviously, we were ready in 2020 for preparing the COVID vaccine. So it shouldn't be that surprising, because there was more than half a century of knowledge accumulated, so we were ready to produce this vaccine. This is just to show you how these nanocapsules made in our lab look like in the transmission electron microscope. So they have a core and a shell, and you have the liquid there. This is the real physical appearance. So when we put a drop of this liquid into a cell culture, these are cells, and the nucleus is in blue, and the cytoplasm is in red. And when we add a droplet of these nanoparticles containing RNA encoding, this RNA encodes a protein that becomes green. So when we add it to these cells, we see this green spots there, which means that the cells were transfected. So the RNA was well transported to the inner of the cell. So you may be wondering, and Dr. Collin was saying, we need to improve the delivery of these materials. Indeed, we need to improve it. How can we make these nano carriers, these nanoparticles? They can be made of many different biomaterials. Those in COVID vaccine were made of cholesterol, lipids, very simple materials, in fact. But it is not only important the number of materials and the kind of biomaterials that you use for making them, it is also very important how you engineer them. In these photos, which are cryo-TM, transmission electronic microscopy, you see kind of layers which represent basically RNA molecules being packed together. So the way you pack these molecules in these nanocarriers is essential for them to work and to transfect and to produce the protein. So who people are already benefiting from these technologies? I'm giving just 2 examples, but there are many, many more on the market already. So COVID vaccines obviously is the more clear example. We could say that almost the whole planet has benefited from these nanotechnologies, not only RNA, but also nanotechnologies. And there are a number of formulations already on the market for the treatment of rare diseases. This is where we are, but it is also true that there are hundreds of clinical trials dealing with other problems and other pathologies. For example, just for the near future, I mean, lots of people are trying to deliver RNA to the brain. We are working with a big pharma trying to cross the brain-blood barrier and to transport RNA molecules to the brain to deal with Alzheimer's, but also to deal with glioblastoma and other diseases. For example, what you see below the head in red, these are neurons that have been transfected, and this is why they are shown in red. We did an in vivo experiment in which we could see that the nanoparticles were diffusing across the brain and were transfecting the neurons. So the brain area is certainly one of the key areas that we hope will benefit in the future of these technologies. We have also been working with infectious diseases, not only designing vaccines, but also developing new antibiotics based on silencing bacteria, for example. And this is what you see there, bacteria with an RNA already incorporated inside them, so to impair their functionality. And of course, cancer. Cancer, you have probably heard about all cell therapies that are already in clinical trials. And what is the relation between cell therapies and RNA delivery? There is a total correlation because these cells have to be modified with a nanoparticle, with a carrier, with something that allows you to change the cell. So nanotechnology is also there. Obviously, gene therapies, silencing RNA, and cancer vaccines. As you know, there are a few cancer vaccines already in clinical trials. And there are many other applications. We are also working on theranostics, trying to diagnose and treat brain diseases simultaneously. And I guess this is all I wanted to tell you today. So ready for any question you may have.